So we started with a very simple hypothesis, which is that TLR4 signaling is important for intestinal inflammation. And so we reasoned that because we were studying innate immunity, we wanted to study an animal model that really didn't necessarily require T and B cells, that was dependent on bacteria, and that was something early, not like one of these chronic models, because we reasoned that innate immunity maybe played a role in the early stages of IBD or colitis in a mouse, and not the later stages. So for that reason, we decided to use DSS, which, at the time we didn't really know that much about animal models, so we started with that. And as you may know from other lectures that you've been to, Dextran sodium sulfate is, I don't know, like a soap or something that they drink. And for some reason, it disrupts the epithelial barrier just of the colon. I always say for some reason because it goes down their entire intestinal tract, but it seems only the colon to be affected. It really wreaks havoc on the epithelial lining, and this results in colitis. So, and it's dependent on bacteria if it's ameliorated by antibiotics, etc. So this is the model we chose to examine. And so I just took a long time to explain to you that TLR4 recognizes LPS. There's this adapter molecule, MYD88, that is important for lots of TLRs. So when we started these experiments and we used a knockout of these pathways, the prediction might have been that these animals would be protected against colitis, right? They don't recognize bacteria, so maybe they don't know there's a problem, okay? The answer is both yes and no. The animals are actually sicker in the absence of TLR4 and MYD88. This is one representation of sicker that I can get away with showing to a GI audience. The animals bleed significantly more when they're knocked out for 4 or MYD88. And you can see that based on their hemoglobins, that their mean hemoglobin drops significantly. We did a lot of work to understand why that was. So they have increased rectal bleeding, fine. If you go a little too long with the DSS, you kill them more often than you would a wild-type mouse. But in terms of inflammation, technically speaking, actually, they have less inflammation. They're not able to recruit neutrophils to the intestine. They're not able to recruit macrophages as well to the intestine. And as a result of this inability to recruit these inflammatory cells, we believe that's the reason why they have bacterial translocation. Bacteria escape the gut and are found in mesenteric lymph nodes to a much higher extent than they are in the wild-type mice. In addition, they have a very clear defect in epithelial proliferation. So it's a defect that spans from the epithelial monolayer of the gut all the way to the lamina propria infiltrate that occurs in this DSS model. So for simplicity, we can conceptualize that the TLR4 knockout phenotype, at least in the setting of DSS, shows defects in leukocyte recruitment and bacterial clearance, and that maybe is to be expected if you don't have a receptor in the innate immune system. But what was surprising was this epithelial homeostasis and repair defect. And I'll actually focus the majority of the talk on that. So we hypothesized that proliferation and apoptosis in intestinal epithelial cells was regulated by TLR4. This is a proof of just the proliferation part. You'll have to trust me on the apoptosis part, and that's published. This is an animal that's been sacrificed after they've gotten a dose of BRDU, which is a nucleotide that is incorporated into their DNA if it's replicating, and then you can easily come back and use an antibody against it to tag it. So a dark nucleus is the sign of a cell that was proliferating. You can see that before you do anything to a wild-type mouse, they're of course always proliferating cells at the base of the crypt. You give them 7 days of DSS, and you can see that there is an increase, but after they've been through the injury of DSS, in the recovery phase, you can see that there's a very dramatic proliferative response to try to repair that defect that's been inflicted. But look at, by comparison, the TLR4 knockout at, again, the comparable time point at the same magnification. I hope I don't have to convince you they have very perturbed, very decreased proliferation. At baseline, we don't really see, if you don't injure the mouse, the TLR4 knockout intestines look normal. I'm not entirely confident that they don't have some subtle abnormalities, but it's so subtle that at least using, level 1 techniques, we can't detect any abnormality. So, with this slide, I will summarize, a couple years worth of work. to try to understand why it is that these TLR4 knockout mice have defective proliferation. And so what we found was that in the absence of TLR4, when you injure the intestine, you cannot express COX-2. So no TLR4, you can't make COX-2. And COX-2 is essential for the production of prostaglandin E2. Prostaglandin E2 has many complicated things it can do. Among them, it can activate epidermal growth factor receptor signaling in an indirect way by causing the release of EGF-like factors. We happen to be looking at amphoregulin, but there are a couple others that it can do as well. We're confident that this is part of the mechanism because if we give TLR4 knockout mice back PGE2, we can restore them back to a wild-type phenotype. They proliferate, and we can bypass the defect by giving them exogenous PGE2. Another way in which this might be working as well is that maybe it's also the macrophages cooperating. You know, the lamina propria is rich in macrophages. These macrophages could also be TLR4 expressing, respond to PAMPs, and they can then activate COX-2 and make PGE2, and still the epithelial cell could benefit from this in a kind of a trans way, not in cis, but in trans. Okay, so all of this led us to hypothesize, well, if TLR4 is important for all of these processes involved in expression of COX-2, EGFR, and proliferation, ergo, this might also be involved in the extreme of that, which is the development of cancer. And so we hypothesized that TLR4 signaling is important for colitis-associated cancer, as distinct from sporadic cancer, which is far more common in the population. And so this is kind of a, an example of that. Unlike the idea that in sporadic colon cancer, when Tim spends the rest of his day in the endoscopy and he's looking for a polyp, sometimes raised, sometimes flat, that he'll remove, and for all intents and purposes, that person is cured. IBD must be different. It looks different. These areas that go on to become cancerous, we believe, are relatively flat, probably not completely flat, but they certainly don't become broccoli sprouts and are easy to remove. And in fact, that has been also demonstrated in the genetic events that occur with P53 and KRAS being much earlier in the cascade and colitis-associated cancer, and APC being a late event. So we were interested whether or not TLRs in general are superimposed on this colitis-associated cancer pathway. Are they involved in initiating events like reactive oxygen generation and things that will mutate the DNA and make it more likely that you'll develop a monoclonal proliferating population of cells? Or does it work later? Once you already have dysplasia, maybe it allows, helps in the growth of that already present tumor. So the first thing we did was we asked the question, is TLR overexpressed in colitis-associated cancers and in ulcerative colitis? And so to answer this, I think it's dark enough. At this time of the morning with not enough coffee, I'd be reluctant to turn the lights down any further. But for the most part, you can see on the left that normal colon, this happens to be a completely normal person, just has some TLR4 expressing cells at the base of the crypts, and Scott Plevy has thought that these are maybe enterochromaffin cells. Whereas you can see next to it is a colitis-associated cancer, so cancer from two different UC patients. The TLR4 expression seems to be much higher. Obviously, I'm talking to a sophisticated audience, and immunohistochemistry and immunofluorescence can be problematic. How do you really know that this is what you're staining for? And remember, this is archived tissue, so there are all sorts of things that go along with dealing with archived tissue. So at Mount Sinai, we were lucky enough to have frozen tissue from a colitis cancer and then surrounding mucosa. So this is a UC patient. The surrounding mucosa is not cancerous, but it's also not completely normal. And you can see on a Western blot, and this is the right size for TLR4, that the tumor expressed much more TLR4 than the surrounding tissue. So at least at the moment, I would say we've probably done about 50 UC cancers, and it's about 80% of them have a high expression of TLR4, whereas it's much, much lower in sporadic cancers.